Immunoglobulin M, or IgM for short, is one of the 5 subtypes of antibodies, a protein produced by B cells to help fight infections. IgM is the default antibody type produced and plays an important role in activating the complement cascade. In this visual mnemonic, I'll give you an easy way to remember all the key facts about IgM. Welcome to the M's Store, purveyor of the world-famous M's candy. Take a closer look at that big M's dispenser. See how the dispenser has its hands up so that it looks like the Y shape of an antibody? This antibody-shaped M dispenser is our anchor for immunoglobulin M, or IgM. Get it? The Y shape of the dispenser makes me think of antibodies or immunoglobulins, and the M's candy represents, well, the letter M, of course. Put this together, and you get IgM. IgM is one of the 5 major types of antibodies, or immunoglobulins. Let's explore the rest of this scene to learn what makes IgM different than other antibodies. First, take a look at that poster in the back. Because these M's are so popular, there are a ton of knockoffs. This store wants to emphasize that the M's sold here are authentic, original M's. You've seen this original branding on the candy wrappers before, right? Coincidentally, this original M's branding helps me remember that IgM is the archetypal or original antibody produced by B cells. It is the antibody type that makes up the membrane-bound B cell receptor, which binds to an antigen during B cell activation. It is also the first or default type of antibody produced by B cells, without class switching. You see, in order to produce other antibody types, B cells need to undergo class switching from IgM to the other types. We cover class switching in our dedicated video on B cell activation. But for now, just know that IgM does not require any class switching to make. It's the original. Is something wrong with the dispenser? It looks like the M's candies are spilling out everywhere. And you know what happens when free candy is around? Kids come running, and they come fast. I mean, just look at how fast these kids are coming. By the way, these kids running over fast symbolizes how IgM is the first and fastest antibody type made when a B cell is exposed to an antigen. We've already talked about why this is. IgM is the original antibody and doesn't require class switching to produce. This is important in the initial stages of a new infection, since IgM constitutes the first or fast spike of antibodies produced in a humoral response, while the second spike, or delayed humoral response, is dominated by other antibodies like IgG. Just remember these kids running over fast to pick up the M's, and you'll remember that IgM responds fast. Next, notice how the kid in the green shirt is holding his 5 fingers up. Why, that's the infamous 5-second rule for picking up food on the floor. That's probably why those other kids are running over so fast, to pick up the M's before 5 seconds are up. Coincidentally, this 5-second rule for M's is a symbol for how IgM antibodies are pentameric. Penta is the Greek root for 5, and mer is the Greek root for part. Putting this together, a pentamer is something that is composed of 5 parts. In the context of IgM, this refers to how IgM usually circulates as a pentamer. That is, as 5 IgM antibodies whose Fc regions are connected to each other via J or joining chains, forming a circular structure. If you want another visual peg to remember this fact, you can picture that 5-pointed snowflake growing on the window. Speaking of the window, see that kid stuck outside the window in the cold? Yep, he's longingly gazing at the candy and the happy kids inside. I just want to say that this kid stuck outside the window helps me remember that IgM is unable to cross the placenta. You see, the bulky pentameric structure of IgM makes it too large to cross the placenta. Contrast this against the monomeric or single antibody form of IgG, which can cross the placenta. You should check out our video on IgG to learn more about why this fact is so important. For now, just remember this kid stuck outside to remember that IgM cannot cross the placenta. Lastly, take a look at that computer over at the checkout counter. Every candy store has a computer for checkout, right? Computers are our recurring symbol for the complement system. You know, since computer sounds like complement. And we're talking about complement here to emphasize that IgM is one of the best antibodies for fixing complement. In other words, the Fc region of IgM is really good at binding to complement proteins to trigger complement activation, which occurs via the classical pathway of complement activation. One of the reasons why IgM is so good at fixing complement is because two adjacent Fc regions of antibodies are needed for the C1 complement protein to bind. Since IgM circulates as a pentamer, it provides 5 Fc regions in close proximity. This naturally produces an ideal environment for complement fixation. I don't want to get too far into the weeds here. We actually have a playlist dedicated to the complement system. For now, I want to contextualize the bigger picture. IgM binding to things therefore induces complement-mediated attack, since complement culminates in cell death via the membrane attack complex, as well as inflammation through the release of inflammatory mediators like C5a. Just picture the computer at the checkout counter of our M store, and you'll be set to remember that IgM excels at fixing complement. Alright, that's all for IgM. Let's summarize. IgM is one of the 5 major subtypes of antibodies produced by B cells. IgM is the default or original antibody type produced and does not require class switching to be produced. As such, it is the first and fastest antibody to be made in response to an infection. IgM circulates as a pentamer, which impairs its ability to cross the placenta. However, this pentameric structure makes IgM an excellent fixer of complement, triggering the classical pathway of complement activation. Okay, now we're actually done with IgM. Before moving on to the next one, let's enjoy some of those delicious M's.